Buenos días
Good morning,
a todos, bienvenidos
everyone.
a
Welcome to this
sesión 4 del Foro
fourth
Virtual Internacional
session
de Tuberculosis.
for the
Esta
Tuberculosis Virtual International Forum.
información
This
se
fourth
tendrá acceso los
session,
analistas y asistentes.
we remind
Está
you
siendo
all that
grabada
it
y
is being
transmitida
recorded and
por
it's
Zoom
being
y
streamed
simultáneamente
via
por
Zoom
Facebook
and
y el canal de
simultaneously
Imagen TV.
through Facebook,
Les recordamos que este
YouTube,
evento cuenta con
and
una
the
interpretación de
UABC
lengua
channel.
de señas
We
mexicana.
have we have
Nosotros
sign
tenemos el honor de ser sus
interpretation,
asesores. Los intérpretes
Mexican
de
sign
esta sesión se encontrarán
interpretation.
en lenguas
Our
moderadas
interpreters
Diana Vianney
are
y Raza
Diana
Rosales.
Vianney, Raza Rosales,
Y
and
la traducción Lucas Nash Everest Muñiz.
Lucas Nash
Y
Everest
se va a estar
Muniz.
transmitiendo
And we're also going to be
por una de
streaming
las redes de YouTube,
through
que lleva
one of the
por
YouTube
ilustración
networks
del
in
grupo UCMEC.
English by
Se
Luisa
cuenta
Diaz
con
de Leon
diferentes
from
canales
OCMA Group.
de redes
We
sociales,
have
tanto YouTube
different
como Facebook,
social
para que
media
puedan
through YouTube
visualizar las transmisiones
and media to visualize our
de
transmissions.
emergencia de UABC.
And
La
you can
transmisión
also access
de
to
este
these
evento
through
está siendo
Imagen
realizada
TV
con el apoyo
UABC.
del Centro
This
de Estudios
event is possible
Internacionales,
thanks
Audiovisual
to
CEPA,
the
Dora
audiovisual
Luz-Flores
services from
y Eduardo
CEPA, who
González-Fagoaga.
we kindly
Este
thank
foro
for
se
the
está
work they have
realizando
done. This
con apoyo de
forum is
diferentes
being also done
instituciones
with the support of different
educativas
institutions,
a través del programa de especialización en casa
Science
de la
Health
Universidad
School
Autónoma de
from
México,
the Autonomous
Dora
University
Luz-Fronteriza, México-Estados Unidos,
in
la Red
Baja California,
Internacional de Salud Fronteriza,
US Mexico
Interdisciplinaria
Border
de
Health
Investigación en
Commission,
Tuberculosis, el Centro
uh,
Nacional de
TV
Programas
Research,
Preventivos y Control de Enfermedades, CENAPRESE, de
preventive
la Secretaría de Salud, Gobierno de
programs,
México,
the National
la
Preventive
Plataforma de
Program,
Investigación para la
and the
Prevención y
National
Control
Network
de la
of
Tuberculosis.
Research and
Eh,
Prevention of
es
Tuberculosis in
importante
Mexico.
destacar
It
que el
is
objetivo
important
del
to
foro es el
point out
generar
that the
un
objective of
acceso
this forum is to
a la
generate an
investigación
access
científica
channel
y la colaboración biomédica a nivel internacional
to
como
innovative sciences with
diagnóstico y
regards to
tratamiento de la
prevention,
tuberculosis
diagnosis,
bajo el
and
nuevo
treatment
escenario
for
de
tuberculosis
COVID-19
under
y
the
para
new
estar
scenario
preparados
of
para
COVID-19
una
and
pandemia.
in order to be
Les
prepared
recordamos a quienes siguen
for
esta transmisión
any
que
possible
tendrán
pandemics.
constancias
We're
para el
going
financiamiento
to
de
give
sesiones
out
y durante la transmisión
certificates for those of you who
se va a
participate
estar
in this forum,
publicando
and
un
we're going
código
to be
QR
publishing a
y un link
QR code as well as a
para
link
generar
so
el
that you
registro
can have access to the
y
registry
asistencia de
so that you
sesiones.
can register your assistance to
A
these
continuación,
sessions. Now,
pasará nuestro
I'm
compañero en
going to
ciencias
pass it
del
on
grupo
to
UABC,
our
profesor de ciencias de la salud,
professor
que
from
hoy
the
estará
Health
moderando
Sciences
esta
School,
sesión.
who is going to be our moderator for this session.
Gracias.
Professor Ricardo Perea, please go ahead. Today we have three speakers, Dr. Zenteno Cuevas and Damián Pérez Martínez from the Public Health Department in Veracruz, as well as Dr. Cuauhtémoc Licona Casani from the Superior Studies from Nuevo León in Mexico. We ask our speakers to please speak slowly so that our various interpreter can interpret your message adequately. We remind you that you have 45 minutes for your talk, and at the end, we should have a Q&A session to give opportunity for those who are following us through social media to participate on this session. Dr. Damián Pérez from the University of Veracruz is going
Recording
to
in progress.
start with his talk, which is the diabetes influence on tuberculosis. Go ahead, doctor, and welcome. Thank you, Ricardo. I don't know if you would allow me to, no, if you would be so kind as to share my presentation, please. I'm trying to share my screen and I cannot do it. Ready. Go ahead, doctor. Oh, okay, thank you. Okay, there we go. Can you see my screen now? Can you see it? Yes, even though it seems a little cut out, it's like half screen. We can only see half of your screen. Is this better now? Yes. Okay, well, thank you. I am very happy to participate with you. This time, Damián is going to join me so that we can share some of the advances we have in some of the research that we have done. This is the content that I'm going to encompass through my presentation. We're going to talk about TB-DM2, then we're going to talk about the effect or DM2 in TB, and specifically on resistance generation. We're also going to talk about the molecular mechanisms which generate resistance in tuberculosis specifically. And then at the end, we're going to present the preliminary genomic analysis on resistant genes and DNA repairs. As far as recent history on binomial, we started our first systematic revision in 2007, and by 2014, we started doing more studies, which presented more data on this raising problem. And the World Organization for Diabetes started working with us. And on 2015, we have the Bali Statement and then the Union Meeting, which is a Congress done through the International Tuberculosis Unions. And the Union Meeting recognized this as one of the most important things to observe. Now, TB-DM is important for the future that needs an attention and a response immediately globally. It establishes that comorbidity goes or fluctuates from 16 to 46%, especially in countries in Asia. And there are 6 countries that have a high TB burden, which will present the biggest numbers, especially India and China. And in the case of Latin America, Mexico is at the top of the list. Now, and we're also the leader in Latin America, followed by Brazil and Peru. As far as the proposals that are established from this statement is that we have to do screenings for patients with tuberculosis and diabetes, and also screenings or diagnosis of tuberculosis in order to follow up on comorbidity or the prevalence of tuberculosis. And it has to be a collaboration among the different health services that are specialists in infectious diseases. And we need, of course, economical resources in order to prevent the impact that these diseases may have in the different countries. Now, right now, all of the impact that we are facing due to COVID is making this, making us have been a little distracted from what's going on with TB. We know that diabetes increases the risk to develop TB, and it also increases 1.5 to 3 times the risk to develop drug resistance anti-TB. Now, the risk is differed according to the different population groups, but the Latin population is one of the most affected. As far as how our epidemiological data are in Mexico, these are the data. And we calculate that we have 422 million people with DM, we have 10.5 million people with TB, and we have 1.5 million deaths. And we have 1.5 million deaths due to diabetes. As far as comorbidity goes, as I was mentioning before, the rate fluctuates between 16 and 46% depending on the country. As far as Mexico goes, 8.6 million new cases were reported a couple years ago. And as far as TB, we have, sorry, for 2019, we had 18,160 cases. For 2020, this is the general scenario as far as the diseases, and I couldn't get any data on comorbidity. In Mexico, we don't have data available. The same goes for drug resistance in TB and diabetes at a national level. I could not find any information, even though I did search a lot, but this is what I present to you right now. As far as comorbidity, there are some data as far as how we're doing. The main disease associated to TB is diabetes, and we're over HIV/AIDS. We have been a constant increase in diabetes in the population that already suffers from tuberculosis. We've considered all of the data that I have talked to you about that has to do with drug resistance. Now, this is a chart that shows the different studies at the different zones where reports were made. This is an interesting chart with all of the information, which shows some important numbers of comorbidities that go from 16 to 35, 36%, depending on the region. In Veracruz, we did this report, and we have a 35.56% on comorbidity, but these are worrying numbers. Because that indicates that a big number of people who suffer from tuberculosis also suffer from diabetes, and this has to do to multi-drug resistance. These are some data that we have at a global level. A person who suffers diabetes and then suffers tuberculosis has a greater risk of developing drug resistance. And in the case of a study that was done in Veracruz, this goes up in multi-drug resistance. We have a very high rate, and we also conducted another study that that showed that 3.5% has a greater risk as far as rifampicin. And we also discovered that most of our patients have a delay on the resolution of the infection, and which goes until after the third month under treatment. And there's a modification of the clinic presentation, and atypic radiologies are presented in these patients, as I was telling you a moment ago. Well, as far as molecular terms and how we're doing on this process, we understand that the drug resistance is because of external stimuli by an antibiotic, and this generates mutations that have to do with resistance, and this generates the resistance of tuberculosis, and this has to do with modification in the body. There's a process where activates stress, and this increases the number of mutations on the DNA, and without a specific order. And what this does, it activates a series of genes that have to do with several important processes for the bacteria. that has to do with flow pumps activation, which allows a less concentration, and this guarantees a longer bacterial lifespan. This is identifies those mutations that may have or may play a factor in order to go back to the original base. So the bacteria generates a relaxing on this remission state. So all of this process in the long run allows for mutations to be generated, which have to do with drug resistance. And this turns the tuberculosis into a drug resistance type tuberculosis. Then we also have this other process under which the internal environment plays an important role. And there have been different reports where it has been shown that certain mutations generate drug resistance on specific drugs, and there are different studies that have showed that this increases DNA mutations, and all of this complex process also has, or it is also affected by these immunological and metabolic alterations that are very likely among people who have modified immune systems, and these because of the antibiotic use, and these results on comorbidity with HIV and diabetes type 2, and this also impacts so that all of these mechanisms are activated and generate mutations that have to do with drug resistance. And this guarantees in a long run for the tuberculosis to survive. Now, there are different elements that are participating under this process, and currently we also have the possibility of studying complete genomes, and thanks to this, we can determine the rate of mutation generation as well as identify the different variants to come out with, in order to have a better knowledge of what microbacteria does, and then follow up in order to determine how the bacteria evolves according to the treatment that it has been given related to the conditions that I have mentioned before. So if we have a patient who suffers from HIV, we can also screen him for tuberculosis, and then we can follow up to see how this patient responds to treatment. For example, this is a normal patient without any comorbidities, and it shows that after this sequencing, this patient starts building the history of all of these here that generate resistance to different drugs. In this case, by day 8, they're generating this polymorph, this resistance to 2 different drugs. So by week 8, this patient is multi-drug resistant. So thanks to this, we came up with the idea of evaluating how we can identify tuberculosis, how diabetes and tuberculosis influence in relation to the genes that have to do with drug resistance. in general terms, what we are interested in is to see how diabetes behaves and how it comes to generate drug resistance. So we don't know how a diabetes patients could have influence with what causes drug resistance in tuberculosis. So then we conducted an analysis with patients with diabetes, tuberculosis, as well as drug resistance, and this study was started this year. It was a bit complex precisely because we had to look for the genomes that we had to look, and Spain facilitated some of these genomes, and we started working with type 4 genomes, and there's a global distribution, and this generates an interesting space for us to work with. And apparently this lineage does not generate drug resistance. So we included all of these genomes into our studies. We work with 4 groups. In people with sensible TB, with resistant TB, with TB, with sensible diabetes and TB, as well as patients with diabetes and TBs, which were drug resistant. Finally, we made a comparison between the different books. Now I'm going to present to you the preliminary results from these genomes which were analyzed. We analyzed approximately 399 genomes from 10 different countries. This shows the different groups. We were a little sharp on the group with genomes of TB. And this is because we didn't have enough metadata from the different patients. Because in the case of tuberculosis, we don't have a lot of information. So it seems that like not a lot of attention is being paid to these kind of individuals. So what we finally analyzed were 43 genes associated to drug resistance. In this sense, we found 584 variants within all of the genomes that we worked with. 167 genes associated with flow pumps, and we found 1,842 SNPs and 65 genes of damage repair. These are the different variants that we identified. The genomes were of different countries, and all of these genomes are real. And what caught my attention was the number of SNPs. Most of them, totally came from the same lineage. So the number of variants called our attention a little bit, considering that that bacteria normally is more constant on keeping its genome. Now I'm going to show you what we found as far as the genes that have to do with drug resistance among the different groups, among tuberculosis and diabetes, and only tuberculosis and drug resistance. And unfortunately, we did not find differences. It seemed like there is not polymorphisms showing that they generate drug resistance. It would seem like the final process of polymorphisms is the same, comes down to the same answer. So we're looking at, for example, in the case of isoniazid, The change to 351 was one of the most abundant ones. This is the common report. In the case of rifampicin, with 450L, we found a frequency of 58, and we did not find any polymorphisms except for a few cases with patients with diabetes. So we did this at a level of sublineages, and it seemed like the response that we get as far as polymorphism that has to do with resistance does not have any influence, or it doesn't have any changes. So we're still working and analyzing this information to see if we can find anything else, and we would comment on this if we come up to any differences among these two groups. As far as the genes that have to do with flowing pumps, everything in blue are the different variants, and what we see down here are the genes, and up here what we are seeing here is each one of the patients that we treated, which in this case, on the top we have non-diabetic patients, and at the bottom we have diabetic patients. On this side, we have all of the polymorphisms that we've found on the two groups, and as you can see some blank spaces, but right here you could see the common presence of different polymorphisms. When we selected only those genes that have important difference among groups, we only found 127 genes, and we could start identifying something there. Ones that we selected a bigger restriction as far as genes, with over 10% among the groups. We found, for example, under this group, which corresponds to non-diabetic, and this corresponds to diabetic patients, and we found this whole here with without polymorphisms, but with diabetic patients, we did find polymorphisms. So this is weird because diabetic patients do not show polymorphisms, but there is an important presence among all of these genes as far as polymorphisms go. So what is the meaning of this different group right here and this group right here with polymorphisms? We do not know. So we're working on this. We're analyzing all of genes. We're trying to find out what its functions are to see if there's a correlation between these two groups. So we continue working. I'm showing you patients, non-diabetic patients, sorry, and we're separating diabetic patients with resistance and without resistance to see if we can find any specific differences as far as this information goes. And this is what we've been working on as far as gene studies. In particular, now I'm going to show you the work that Damián has been working on, which is a little bit more advanced, and I'm going to stop sharing my screen so that Damián can talk to you about this data right here. So Damián, are you there? Yes, doctor, thank you so much. Okay, well, just a friendly reminder for our speakers to try to speak a little slower so that all of our interpreters can interpret your message accordingly. Okay, continuing with the same narrative, we were analyzing the genes that repair harm in tuberculosis because they have been observation focused in the last years, and it has been found that there are more polymorphisms with the genes that repair damage compared to constitutive genes. And we know that tuberculosis is a very resistant bacteria, and part of that resistance is thanks to that system of repairment. Some lineages, such as the Beijing one, has polymorphisms that made it more adaptable to generate drug resistance. On top of that, there are different ways of repair that overlap, which speaks about redundant functions that keep maintaining the integrity of DNA, even under conditions which are difficult for the bacteria, constant harm of the immune system, drugs, among other things. And we also thought that Mycobacterium did not have a system of adjustment repairs. However, some studies have shown that there is a gene that covers that function. Now, this gives it genetic stability as well as diversity. And many studies have taken place, and 65 genes have been focalized that are related to DNA harming tuberculosis. However, we don't know the influence that diabetes might be having on these genes. In a very general way, it is important to know the different routes that the microbacteria use in order to repair their harms. When a base, here on the left, when a base is damaged, it can be for different agents, it can be because of a substitution. This harm or alteration is scanned by certain proteins which start different routes, whether it is a restore route under which one base is eliminated or a group of bases is eliminated in order to correct the damage. We also have double ruptures on the DNA chain, which also have their own repair routes. We have repair through under one second way to repair these. However, when the chain is far away from this route, it can use other routes such as the union at the end. Now, these two are mutagenic because they're answers which the responses who do not control the genetic content, and they just look to keep stability. As Dr. Roberto was saying, we collected 399 sequences, which were processed at the Biomedicine Institute in Valencia through cleaning first so that we could have quality control on the polymorphisms that we observed, making sure that they wouldn't be contaminated with DNA from other species. Then we conducted something that's called a variant with tuberculosis complex, which was designed by Dr. Ejaki and his collaborators. And we identified the presence of polymorphisms which are related to drug resistance. We also confirmed that the genomes were part of lineage 4 and also the sublineage was identified. We recognized two big variants, one of it being fixed variants with over 90% of variants, but we also have a different variant group, which is known as not fixed, which was a little bit over 10% of our genomes. We have to recognize that these non-fixed variants are the changes that are more common among the bacteria population among our patients. And it would seem that these would prevail in the dominant population. For now, we have analyzed the fixed variants to identify the genes that have to do with harm repair. We analyzed and all of these data, and the first thing that we were faced with was that 65 genes analyzed were identified with 534 polymorphisms, but they were also polymorphisms that conducted to a great number of polymorphisms. 70.4% were unique. In other words, they did not match any other genomes from our sampling, and only 5% had a consistent frequency. That's why this analysis is at a gene level with polymorphisms. These 65 genes were analyzed to see which had the biggest frequency of polymorphisms. And we observed that some genes had over 15 polymorphisms, most of them DNA2, but we also found some genes that have a very low frequency of polymorphisms, and we realized that the smallest the gene, the less polymorphisms. Now, the gene RBUX, which lacks polymorphisms, has structures of branch DNA, which will represent the, if it's not solved, it would mean the dead for the bacteria. Now, as far as patients with diabetes and without diabetes, we observe a lot of important aspects. For example, we have the patients with diabetes, and at the bottom, patients without diabetes. There are genes that have polymorphisms that are related to the lineage, and on the right, we have the genes that are covered by these red marks, and these are genes that do not have polymorphisms unless they are on patients with diabetes. And right here, we would find what we would identify as the patient mark. And we're identified that patients with diabetes mellitus have a higher risk of generating different kinds of harms, and they could be creating this diversification of the genes of harm repairment. We identified that a gene with a polymorphism could change the function of this gene, making it more efficient. But it is also possible for this verification to respond to the need or the advantage to stop the function of the gene. We have other polymorphisms which are indicated with these arrows, which are only present in sensible strains from the different groups. Now, this is the repair system before being selected or registered by the antibiotic. Now, on the central part, we found a distribution of the polymorphisms in the genes, which varies a little bit more, but we can also observe red lines on the two groups, which are related to resistant genomes. And we can see some of them, such as UVRA, where polymorphisms are bigger. In this sense, we identified 6 genes which were statistically important as far as the difference between the hosts or the different patients. These 6 genes have the next functions. We observed that MUD-M, FPG were implicated in the oxidated guanine, which is the base for the most damaged DNA. And when it had mutations with these two, and being related to the resistant strains, it would seem like they improve their functions too. Also, UVRA showed a greater polymorphism burden, and it would start repairment through an NER. And DNA B together with DNA A initiated repair with a double DNA change by an dependent HR of Reg A. Now, this is important when production of different genes start being productive, such as polymerases, which can generate mutations on the genes. We also found mutation in some ligase, and we have a little bit more of information that's related to drug resistance. This gene takes care of the DNA structure in the stationary phases of the bacteria. And it has also been identified that the presence of polymorphisms in REG-GWED is a link for the strains 421. And we can see this could indicate that this is the result of the positive indication of the treatment, which gives the bacteria an advantage, and it affects the immune system. Through comparisons between the different hosts, we also found a difference among the polymorphisms. Among these ones, we found that the RCA gene is more frequent in diabetes mellitus host, on sensible isolates as well as non-isolates. Now, this could indicate that diabetes is having an influence on the development of polymorphisms, which also are found in different isolates. The major frequency on polymorphisms could explain the risk that these hosts have to generate drug resistance. And this is a question that we are interested in answering, but we're limited because of the studies that we have been able to do, but it has given us some guidelines for future research. Through these polymorphisms, we came up with a phylogenetic tree to see if we could find differences between the different hosts throughout this distribution. However, we could not find anything to the answer that we have through these are the different sublineages that are compound, that the sample is compound by. We found the strain of the 4.3 lineage, which is completely different from the rest of the groups. Then we selected a group of 18 genes through which we could support the classification of the sublineages. And we generated a smaller tree, which is a functional one. These 24 polymorphisms are distributed among 18 genes, which are the following. We found diverse function among which we found a lot of ligases. We also found a lot of them with OGT, ADAB, DNA E2, and we also found REG-GWED, which was related to drug resistance, and this takes us to think that the different lineages have associated mutations that adapted, and we don't know what its potential can be when all of these polymorphisms get together from the different sublineages. Something that allowed for us to do through the polymorphism diversity is for us to come up with some discriminant models. After identifying the differences among hosts, as well as among the lineages, we conducted a discriminant analysis, which allowed for us to identify 73% of the strains that came from diabetes host. And through multiplying the presence of polymorphism by the volume of the coefficient of the gene, we could do a simple addition, and we could come up and we could see that the host with that strain was a host with diabetes. And we could also identify a host without a diabetes. This model was tested according to the different methodologies, and it showed us a classification of a correct classification of 70.2%. However, we are still pending to improve this model, and we have to continue studying the polymorphisms that are associated to the diversification to the host related to the sublineages. An important aspect of our discriminant analysis in order to identify hosts with diabetes is that we were able to identify over 25 polymorphisms. With these 25, we were able to come up with an important model, and statistically, this was discriminant as well as discriminant in average. For the isolated with drug resistance, the model was performed with 64 genes. We only got rid of one, which did not went over the necessary limits for the statistical test. Now, under this, we classified from 70 to 80%, and it was utilized the same way, multiplying the presence of polymorphisms by the gene volume, and then you would add the next polymorphism by the gene volume. And both models are functional, but we need to still conduct some tests before we can start proposing them as useful tools. Now, something important is that in the validation bed cross sampling, one of the registers was taken away so that we could analyze it apart from the rest of them. In this case, 38 registrations, and we came up with a result that decreases the accuracy of our test. That's because the N is very small, and when you get rid of one registration, this fluctuates the percentages, but we still have a 70% cross validation for sensible and resistant traits for diabetes hosts. And this is giving us signals that this is a possible route that we can follow in order to come up with a useful model. As far as conclusions, well, this is the first analysis work under which we analyze the polymorphisms present in the context of the binomial. And we were able to show the genes with patients with and without diabetes. And it also has given us evidence that the microenvironment of the patient with diabetes is influencing the diversification of the genes, the harm repair genes. And we're under the process of ratifying some results. We're going to use the most variable polymorphisms in a bigger frequency, but we also need, of course, to use other research models such as the longitudinal models, which will clear um, the overview as far as these tests go. Doctor, thank you, Damián. Thank you so much. Now, this is a preliminary study. We have still some points that we have to analyze. We have not found now the final response could be the same. Now we can identify in the presence and absence of polymorphisms and it's something that we've been analyzing. And as far as repair genes, there's a high rate for the development of drug resistance in patients with diabetes. We should have a study of series for those with diabetes who also develop tuberculosis so that we could calculate a mutation rate to see the speed under which these changes happen. Now, if you have any patients with diabetes or patients who have these comorbidities, they would be patients that would be very welcome to be included for this study that we're doing so that we could sequence the genomes and we could conduct the analysis. So next slide, Damián, please. Okay, well, now I would just like to thank my colleagues from the Biomedicine Institute in Valencia, Dr. Carlos Madrazo and Irving Cancino, who were very helpful with the preliminary analysis for the genomes, Dr. Betsaida Cuevas, who has also been helping us with the analysis of the polymorphisms in flow bumps, as well as Master Gustavo Bermudez, and Damián, who's working or who has been working on DNA repair genes, as well as Diana Viveros, who has also played an important part for this research. And I would also like to thank CONACYT, which is funding a project which has been a little bit delayed because or due to the pandemic, but I insist if you have these kinds of sample, we would gladly welcome them so that we could use them for our projects and research. Thank you so much. And you can find my email and you can contact me anytime for any questions or comments that you may have on my presentation. Thank you so much for your presentation. We have some questions from our audience. And the first one would be for Dr. Zenteno. As far as the relation between tuberculosis and diabetes, could you repeat the question, please? Okay, if there's a consideration of the tuberculosis population to be integrated on the nutritional programs. I'm not informed about that. I think it would make sense. There was a program for patients with diabetes so that we could prevent them from developing tuberculosis. But that part specifically, I apologize to you, but I'm not well informed. So I don't know. What is the recommended frequency to follow up on the sequencing for a patient? As well, there are not specific recommendations. Practically, more or less, you have to understand the way the bacteria works. It's a bacteria that grows slowly and it duplicates every 24 hours. So it would be wise to generate a sample every 2 weeks in order to evaluate the evolution of these bacteria. I don't know if you have any additional data, Damián, that you could share. No, doctor? Well, it is important how deep you do this, how deep you go on your sequencing in order to have information on the more on polymorphisms with more frequency. But literature marks that from 1 month of treatment to the next, the results are different. So, like I said, between 2 or 3 weeks, you can start seeing variation on the different types of bacteria that could be present in the same individual. Is there a relationship between the drugs and the different schemes that you use with your patients as far as the bacteria response mechanism? I think they're handled the same way, even though literature shows that there are important differences and that bacilloscopy is prolonged after the second or third month. I think that you can handle it the same way. I don't know if there are any differences. I don't think so. As far as the managing the drug management for diabetes patients, and it is important to also have a glycemic control with patients with diabetes. And I don't know if there's a preventive drug treatment. I think it's specified in the different literature, but we know that reality is different. There are different studies that have been shown that there's a direct impact of all of these drugs that are applied to control the sugar level on the blood. There's a very interesting study which was done by Dr. Raquel and yourself, Ricardo, which talk about the different concentration of drugs within patients that suffer from diabetes. And it has been shown that there are differences of the negative influence from some drugs as far as keeping the concentration of drugs which are ideal for tuberculosis patients. So there are some studies about that. Now, why is it necessary to analyze the non-fixed variants? Well, they are necessary so that you can see where this variation on the bacteria is coming from and where its path is going to be. Among these not fixed populations, you can find resistance genes. And if you can find a gene on RPOB, you could think that this colony would evolutionize into a drug resistant bacteria. And it is important to identify this so that we can find the host print on these genes. Well, thank you, Danielle. Now we're going to move on to our next presentation. We would like to remind the speakers that we have in sign interpretation as well as language interpretation. So we kindly remind you to speak slowly so that they can do these, so they can offer this service adequately. And now we have Dr. Cuauhtemoc Licona-Casani from the Tecnológico Institute in Monterrey with the talk, A Cellular Landscape in Tuberculosis. And doctor, if you were so kind as to sharing your screen, we would appreciate it. And the floor is yours. Can you see it now? Yeah, okay. Okay, well, thank you so much, first of all, for the invitation to show what we've been doing with multi-drug resistant at our Technological Institute in Monterrey. This work, I would like to briefly mention that it is a work that we started by the end of 2019 with important collaborations among our institution and the IMSS and the research unit in Zacatecas. And we started doing this because it was our interest to understand beyond identifying what we need to come up with diagnose. It was important for us to understand the metabolic and cellular mechanisms that are present when there's a kind of multi-drug resistant. And we did it on Mycobacterium tuberculosis because it's an important disease that affects our population. I'm going to show you a timeline, which is interesting for me to show to you because this is one of the diseases who has prevailed for many years among us, and I think this information is relevant. This is a disease cycle that has been registered under our health system since 1882. This was the beginning of this cycle. It took about 10 years for a vaccine to be created. And this one of the first vaccines generated. And it took 10 years and the establishment of the first vaccination protocols of, well, the official vaccination protocols. And obviously under this path of 40, 50 years since the vaccine was developed until, well, 20, 30 years until it was established. And even when vaccinations discontinuation or vaccination stopped, due to the barely adapted concepts that were studied about vaccination, such as Lubeck disaster, which has to do with lack of quality control of the vaccine in the highlighted strains. But there were some important advances under the time that was called as the golden age, with the first strains which were producers of these and these different groves, streptomycin, isoniazid, etc., from the establishment of the massive production of these compounds, these small molecules. It is also important to know that ever since the AMR were discovered, there was a boom for these antimicrobial drugs. Now, the resistance phenomenon and some interviews document that there was a misuse of these molecules, which led to drugs resistance. From in about 100 years, we concluded with genome sequencing. A few years prior to this, we started showing that it is important to understand the transmission phenomenon as far as tuberculosis and to take preventive measures. This was recognized by the World Health Organization, and it also, and tuberculosis was also recognized as an important worldwide disease, and the genome was known, and of course understanding the genome totally allowed for us to start to start coming up with more and better detection and prevention techniques. So we started developing a new detection systems, which were very precise compared to what used to be done before. And we also started using some drugs and antibiotics, especially during this age, the post-genomic era. Once this disease became so relevant because of its resistance level, we would like to show you everything that has been invested. Millions of dollars have been invested globally in research. And if we take into consideration the last 25 years, we have an increase or an accumulated of 6.3 billion US dollars. And within this timeline, it was established by the World Health Organization a goal that specified that by the year 2050, we should have eliminated the disease. We should be able to eradicate the disease, because this is a disease that has been modeled a lot, because it's one of the oldest diseases that has prevailed worldwide. And in order to get to this goal, we would need to have an estimated investment of $9 billion on TV R&D studies over the next 5 years to meet the NTV strategies that we are looking for. Now, something that is very relevant is, of course, the consequence of inaction. This is very dangerous and worrisome. For example, a 5-year delay in satisfying the R&D funding targets could result in an additional 8.4 million TB cases and 1.4 million TB deaths by 2030, which is not something that we look forward to. Now, well, the goals that have been set with the money that has been invested, and what we need to do is do more with less. We have to try to improve and come out with more effective vaccines for tuberculosis. We need to generate point-of-care technologies in order to diagnose tuberculosis, etc. All of these branches should be developed in a safer and more effective way. And in order to do this, we have, obviously, through the different advances and developments in genomics, as far as the informatic tools that have been developed, and the abundance on all of the data that we have something that's called genomic epidemiology. And this just has to do with advancing on things such as detection, sampling, and sequencing. We have to try to use these technologies, which have lower costs compared to some years ago. We have to come up with techniques that may enable us to sequence more samples quicker and in a more affordable way. Now, together with these requirements, we also need, obviously, for the analysis tools, the genotypical analysis tools, be more efficient and more general. This is a chart that you are probably familiar with that has to do with all of what I have mentioned before. Now, we all know that the tuberculosis bacteria is a monomorphic bacteria with the limited geographic distributions among individuals of tuberculosis strains are genetically monomorphic, therefore nucleotide identity, it has a nucleotide identity of 99.9%. To identify the size of the problem, all of the differences can be mapped, could be found in a map genetic distance of 2,500 SNPs. Now, the genomic epidemiology and its distribution is shown here in this phylogeographic distribution. We have a very detailed distribution, and even new lineages have been able to be identified. Why? Because we have a genomic density, a microbacteria population density, which is very varied, which allow for us to identify the different lineages. Now, getting to the final point, before I start giving you some data that I want to show with you, the contrast is very important to mention that there's very little variability. There are very little phenotypes. Yet, behavior also has the limited geographic distribution, and but phenotypes vary with regards to the lineages, and one would expect that if we at genome level there's not a lot of variability, this would be the case of the phenotypes, but this is not how it works. That's why it's so important to study and to sequence genomes at a very high density in order to understand what are the cellular mechanisms that determine these changes among these phenotypes. Now, I'm not going to give you more numbers, but the importance of studying these phenotypes, I comment that, and you might already know this, by 2050, two independent models suggest that if AMR is left unchecked, and if we don't continue doing research on this phenomenon, we're going to culminate on a 2.5 to 3% loss. Now, I'm sorry, this is the concept of the talk that I would like to show you. This has to do with the determination that we as users, if we want to call ourselves that, we use in order to determine drug resistance. This is a pipeline or a logical way to work under which a lot of strains have been sequenced worldwide, and correlations have been established. Genotypical changes or variations at a nucleotic level. And all of these are printed at different platforms when we use a genome, and we come up with a forecast. What calls our attention is that if we want to have a global strategy, we need to have these forecast platforms globalized. And unfortunately, the density of sequence genomes in some areas are not a lot compared to others. Obviously, this is because there are some guidelines that you have to follow where there's a bigger incidence compared to other places. But now that we are all here today, I can show you the big problematic that this represents for Latin America. Now, well, this is the density in sequencing in our region, and it is very low, if not completely absent. And these efforts have been very, very little, or we, in many places, we lack these efforts. Even though there are important sequencing exercises, such as the one that Dr. Roberto Zenteno just presented to us and some other groups in Mexico, we really have not gotten to a point where these genomes are a part of these platforms. And in that sense, my lab, the industrial genomics lab, are very interested in understanding and developing these critical mass of genomes that would allow for us to establish a correlationship between genotype and phenotype in Mycobacterium tuberculosis drug-resistant isolates in Mexico. So the concept is, as I was saying, the population density has determined generating some targets, and these targets are very well established in some platforms, but this population density does not represent all of the lineages that we have determined. Now, how can we accomplish what we want? Well, we need to try to do more sequencing in some regions, and we have to try to create some co-relationship through a platform that would allow us to see the perspective of all the nucleotides that have to do with this sequencing. These sequencing are going to allow for us to generate new diagnoses that could be included as part of these platforms, these analysis platforms, and somehow it would allow us for us to globalize all of these analysis so that we can take informed measures in the regions that these are needed in our planet. So an important collaboration was generated between Dr. Antonio Ciso, which is part of the Biomedical Research Unit of Zacatecas, and he got together with Dr. Roberto Zenteno from the Veracruz University to have 133 isolates, some of them which we sequenced, and at the end we have 133 isolates that are that have the genotypes with the resistance, and we generated a common sequencing platform. There's nothing new under the sequencing that we perform, but we are trying to be innovative as far as the analysis platforms that we're generating in order to generate a platform that could be free of access for everyone, that is user-friendly, and that could be useful for people that are working directly at the clinic. So we're working on this with student Paulina Mejia and Axel Ramo, which are very outgoing students. Sorry. So out of the 133 genomes that we have as a case study, we have a variability of lineage. We can see somehow different lineages and sublineages under lineage 4, which are found among this genome population. We have also been able to perform an identification exercise on the transmission processes for some strains that we were able to isolate, and we were able to register their isolation cycle, co-located at a geographical region to determine whether or not they have the requirements at a genome level, in order to determine whether or not it is a process of transmission. I can show you how the different lineages are distributed. We have the X type and the Lam and Carlem, and they're color coded. And I would like for you to observe, we're going to go over later, but we have a registration of the different comorbidities that we have. And we have also some geographical data for each patient, and of course the resistance results. And with the tools that we've been using, which are available for generalized use, we can generate some maps according to the isolates that we have in the distribution that somehow can be seen now, which is aimed a lot more towards a diabetes study in Veracruz. I'm sorry, I lost my train of thought, but we have a distribution, a heavier distribution in Veracruz, and with these tools we can correlate whether or not we have profound data on each sample. But what we're going to do right now, and what we're going to talk about, the objective of our talk is, as far as we're going to focus on the resistance phenomenon, and I show you how we're doing from all of these genomes that we've been analyzing right now. I can show you how they are distributive, sensitive for light blue for sensitive, pre-MDR, a little darker blue. This is what identifies the extremely resistant and MDR, and they're all color coded, and the size of the circle also represents the number of samples, and we're very focused in Veracruz right now, but the idea is to start generalizing these studies, and we want to start inviting people that have the possibility of extracting some isolates to work with us so that we can populate this map from Mexico with a lot of screenings. Now, one of the main things that called our attention when we began doing this, we said, well, we have certain isolates that tells us genotypically or certain tendency to drug resistance, and we run them with platforms that show us we run them through these forecast platforms. Well, this is a chart of distribution. We have 35% of MDR, etc. If we could do mapping for each genotype, if we could do these for each drugs, we could identify where these mutations take place, these variants for each genotype. As of today, well, right now what we're observing is the samples, and these bars show the frequency. Now, a lot of these are canonical genes. These are blank or white genes that are a part of the prediction tools. We are all familiar with these, and we know that this kind of traffic is common because they're the genes that we are all looking for, because they're the genes that have been established as guides for diagnosis. And this is how we have the charts. We have the canonical genes where we can find the mutations, and the bars show the frequency by gene. So obviously we have HR, HRES are the strains that are the which are the most which are the most represented positions. It is also important to know that on this side we can see in one of the in one of the most common platforms, TB Profiler, we can see the results that these samples could show us. And what we can see is the discrepancy between what exists and what's forecasted without us even looking at the phenotype and what we are sampling. In other words, the 35 of our sampling or MDR, we have that really we're looking or we're observing these discrepancies or these variabilities. If this would be 100% agreement, all of these would have to be dark pink. So all of the sampling that we're seeing here are samples that do not agree with what we are observing on our phenotype at the lab compared to what's generated on the forecast. And this is important because this has a lot of implications, especially for those that are sensitive. We send these to sequencing and we can see that there are some samples that are shown as sensitive, but a lot of the sensitive samples show that the genotypes are not happening that way. So we have samples that are extremely resistant. I have a sample that's within a sample that's forecasted as sensitive. Now, the central message here is that there's a discrepancy, and that this discrepancy can have serious implications among the health system. Now, talking about correlating the level of prediction that we have or the level of forecast that we have, we see that for some resistance, we have some correlation values that are very poor. For example, the streptomycin or the ethambutol, we have a very low forecast level, which is somehow a symptom of knowing or it indicates that it is important to generate rules that could be added to the existing rules or guidelines on the forecast platforms that we all use. In other words, they're not very applicable for to the isolate populations, especially for the ones that we have in Mexico. Now, what do we do with this? Well, we have a perspective of the genotype and phenotype drugs resistance, but what do we do with all of these variants that we have at a new nucleotic level? We have a perspective within all of the strains. Each one of these is an isolate which are classified by lineage and by colors and are color coded. Well, by these shades, these colors here show the resistance pattern. And somehow we were probably expecting that a sample which is sensible would have less variables compared to a sample that is extremely resistant or MDR, multi-drug resistant. Well, this graph does not show it very well, but at a level of, not statistic, but at a general level and with the little amount of genomes that we have, we cannot actually see a trend. But what is or what was expected, if we think about this, is that these genotypes have to be expressed at a level of post-transitional events. Anyway, we don't see a direct trend, but with the kind of analysis that we have, we can start to identify, oh, what you are seeing at this type of circles, what you're seeing here are the samples on the left side, the samples that are resistant to isoniazid and rifampicin resistant isolates. And on this side, we have a genomic map for tuberculosis. So for all of these, for example, for this isolate in yellow, we have a series of hundreds of points and lines among the genome where a variant was found. And we do this for each variant, and somehow we can detect if there are any hotspots within the genome where which agree with different strains. But this is not very clear. However, we can perform some functional analysis to try and find where all of these variants are within these 400 genes analyzed. And we could also do this combined analysis with, well, combined as far as the different antibiotics. In this case, I show you strains that were resistant to isoniazid, rifampicin, streptomycin, and pyrazinamide. And all of these strains showed that in the same position or very close to each other, certain variant. And we can also do functional analysis so that we can interpret them and find out what these genes are or what the clustered genes that are more representative. And we can do this for any combination. Some being more relevant than others, depending on the isolates that we have. In this case, we only have 3 isolates from 273 genes that analyzed that have this amount of resistance, and the argument falls over because it doesn't have a lot of different representations. But the idea is to start having some kind of a statistics on the analysis that we have had in order to also be able to compare. Well, anyway, what we wanted to do is from all of these profiles that we have analyzed for multi-drug resistant isolates, we have this number of variants. Now, it is important to highlight that these variants have been filtered. These variants have been filtered by variants that are not present on the sensitive strains. In other words, if we were to compare, or we would perform a Venn diagram and all of the variants that have been seen through the different sensitive samples, if we would classify it as variants that give you not the genotype but the lineage, all of these were taken away from our analysis. We have also taken away the variants that show somehow of effect on the translation that are synonym translations. So we took away all of these variants that have an effect on the interpretation at the end. So with this analysis, we started saying, well, somehow it is too much trying to correlate all of these lines here on the right, because this looks like a complete piece of clothes. So what we started doing with one of our students, Axel, we started a development of an algorithm to perform functionalized variant analysis. And as you can see here, the data structure that we have for this type of analysis is this. We have positions at a genome level, and we also have samples. And we can identify whether the sample had or didn't have a SNP or a mutation. And we can do this for each and every one position that the microbacteria genome has. This way, for each sample, we have an extra matrix which shows demographic information, demographic metadata matrix, which gives comorbidities, ethnicity, drug resistant, age, gender, etc. So what we generated somehow is a tool that can be utilized or which will be soon available to be utilized to do different correlations and to be able to assign functions and then correlate the metabolic or cellular functions that are associated with each position, which is shown here on the left. So each function on each position on the left is going to allow us to do filtrations of the genotypes or phenotypes, I'm sorry. And we are going to be able to do different things. We're going to have a matrix data set with variants to isolates and matrix one at the demographic metadata. We're also going to have functional notation of all the variants detected. We're going to have selection of a parameter, and then we're going to be able to do functional analysis on the different variants for the different isolate. So this is a tool that we now have, and we have even done, well, what we're seeing here is on the left. This is a platform of prefiltrations for microbacterium genome analysis, and we've been using it to do some network visualization analysis. So for each genotype, we can see how these genes are shared or whether or not they share mutations. And we're trying to include all of these to algorithms of deep learning to use these pre-filters of genotypes that we are observing here at the lab. And those mutations that are being generated within all of these samples are going to allow us to inform or generate rules that will help create better algorithms. This is a very interesting process, and we're doing this together with mathematical studies in UNAM, or the Autonomous University of Mexico, and our student excellence. Anyway, one of the things that I would like to show you, just to finish my presentation, or I'm about to finish my presentation, what I want to tell you what's happening. How can we do a functional mapping of the resistance phenotypes? And when I say a functional mapping, is what can we do to identify those genes that show mutations in order to generate a hypothesis as far as the cellular and metabolic cells that are behind all of these. Now, I show you here that we have 3 genotypes, genome populations. And from these phenotypics, we have genes. From these phenotypes, we have genes that are, or that show mutations for each phenotype, sorry. In this case, we have mutations or SNPs, which are exclusive for each phenotype. And these genes are exclusive, or they're exclusive variants for these phenotypes. And that's how we could sample some genes that would show mutations or SNPs on shared phenotypes. And these are the routes to acquire complex phenotypes. Not only that, but the size of each node refers to the frequency. In other words, because we have a subgroup, the P subgroup, the orange subgroup, which represents a population. It represents a certain number of isolates. The size of each node will give us what we call a fitness score, which is equal to the number of positions who were affected by that gene divided by the frequency. In other words, whether or not these positions were repeated in some other isolates. In other words, if we have a fitness score or a vigor fitness score, it means that these are genes that have been or that have mutated or that have shown functional changes in a greater number of strains. So we start we start being able to do this kind of analysis. And one of the analyses that we do is let's try and find a correlation. This is the strain network that are very within nodes that are very resistant. This node is resistance to just one drug. We start doing this mapping and then we start doing a correlation, and we can see the exclusive genotypes for the different drugs have to do with these main functions. And I'm not going to get into detail due to the time that we have left, but we have started developing some hypotheses. And from those hypotheses, we're trying to correlate or do a correlation with the mechanism of residence for each antibiotic to try to interpret and then generate hypotheses as far as this underground mutation generated under in genotype. And we have these for each antibiotic, and we have some observations that are going to be discussed under our soon to come out publication that have been recently working on. Now, in this case, what we are observing is the strains that are resistant to rifampicin, and these strains or these populations that we have genes that are mutated, and they present changes on these main functions. These are the main functions on isoniazid, and we decided to compare them from the strains that are determined clinically are multi-drug resistance, which are the strains that at least have resistance to rifampicin and isoniazid together. We have very few SNPs or very few mutations on genes that are exclusive for antibiotics individually, and that really the type of genotype for multi-drug resistance leaves a very big mark compared to others. On green, we have metabolic guides that are mutated, and in red, we have some genes that are genes of repair that are non-metabolic. Therefore, in these regions that I am marking on the left right here, these gene groups indicate some routes. These routes, these little landscapes or routes that are shown here are routes that we can follow for the acquisition of complete genotypes. Now, the residences of from here to here should be, these routes should be printed in order to generate the complexity of the genotype from one to the other. It is also important to analyze which genes are these and analyze their metabolic background on drug resistance. And I'm about to finish. In summary, what I would like to tell you is that with generating the critical mass, we have a genomic platform for functional analysis of MTB clinical isolates in Mexico. and we are generating sequencing platforms, extraction mechanisms. We have very good collaborators. We have a good team in Mexico, which has given us a lot of samples, and we're also generating functional platforms, like I have mentioned, that allow for us to generate the type of studies that we're interested in. So we are writing an article so that we can start spreading and the tools that we are creating so that other people can use them, so that we can generate this type of analysis, especially, obviously, this cannot be done well. All of the people that are without the help of everyone who is a part of this call today, including Dr. Antonio Enciso from the IMSS in Zacatecas, Julio Castañeda, Roberto Zenteno, students from my lab, from Monterrey, Paulina, Axel, Luz, and a lot of other partners that have given us samples and have shared a lot of data. I would like to thank you all. Also to the task force or work group that's working on microbacterial genome, and we've been working a lot with all of these young people. Thank you all so much. My email and my contact information is right here. And with this, I close, or I finish my presentation, and I would be more than happy to answer any questions that you may have. We do have some questions from the public. And have you thought about implementing this sequencing? And if so, what can you tell us about the time and costs? Implementing the sequencing genomes, do you have any data as far as costs and time? Well, it's one of our main goals as far as genomic epidemiology. It is important to be able to have all of the information on the genomes. Right now, some techniques and experimental procedures have to take place before we get to extraction mechanisms that could be more efficient. As far as sequencing, there are many things that we have to work on. For example, right now, it can take us perhaps 3 weeks or maybe 2 weeks to do sequencing, considering that you have all of the kits that you need and all of this timeline. It is not the best, but we try to do what we can with what we have, and we are we have some barriers that we have to face, and that is translated to our efficiency. Now, the system that you have presented, how is it utilized for the clinic management of tuberculosis? Well, these are the pillars of the analysis, and what I have been showing you is a little bit of the analysis logistics definitions. We have to generate a database. We have to generate algorithms, automated analysis algorithms is what we need. And this is the first part. And obviously what we have thought about is for it to be available to everyone, to any person who may have a genome, whether it is at a clinic or at a health agency, for it to be automated. There are some platforms that are already available, so that where you can upload a genome and then it can tell you the lineage more or less. And this type of tools would be the ideal. Our goal is for them to be available for any person who works at a clinic or at a university or anywhere. We're far from that. We have not gotten to that yet, but that's our goal. Well, as last as the cross-analysis and the resistance network, when this functional analysis agrees with the list of genes, and from what you have shown, go beyond with the when it agrees with the white, the pharmacological white genes. Well, that's an interesting question, because that is precisely one of the weak points on the analysis that we have shown you. We don't have enough genomes. It would be important to do this analysis with a lot more genomes, but I wouldn't like for this to seem like a negative connotation, because from what we have seen, we have been able to find clear discrepancies as far as genotypes, but we obviously see the canonic genes or white genes that are also there and are a part of these gene groups that we have seen with changes. Our focus is not to focus on these 10, 15 genes that we are all familiar with, and that we know that are drug resistance. We need to find others that are resistant signatures for autochthonous genomes, and try to find some kind of interpretation of the mechanisms and for which all of these are correlated, and that information can also be utilized ideally to be utilized for a diagnosis or some kind of decision making for the way that you decide to go. I have this strain that I can see that has maybe not, it's not resistant to a streptomycin, but I am detecting some signatures that show that the path that is following is leading to multi-drug resistance. So these could be useful to determine this. That's what we would like to see. Okay. A predicted strain, if it's sensible under your strategy, the difference between prevention and detection. What can you say about that? Well, yes. This is something important to note, that the phenotype screening done at the lab has certain limits and something resistant thresholds, and you have to do some color-coded analysis, and this is important to know because we're trying to see whether it is resistant or not, but we also have to take into consideration all these limitations. Thank you, Doctor. Another questions with regards to what you were saying as far as the autochthonous genes in Mexico. How have you been doing the characterization of this frequency for these autochthonous lineages? How have you characterized, or what are the autochthonous lineages in Mexico and their correlation with drug resistance? Well, really, we have the expert on this panel, and Roberto would be the person who could better respond to this question, because he's the best prepared person. But it has been detected that there are some lineages that are only found in Mexico, and some of the analyses that have been generated have been allowed for us to determine that there are some that that are of transmission. Genomes require to have a profound analysis, certain quality in the sequencing. It also requires certain things that have been standardized by the community. And with what we have sequenced, which is a lot, all of the efforts that have been done by Roberto and his team are important, and the resistance, the number of genomes that we have is very, very small. So we cannot say that, oh, well, autochthonous genomes did have this resistance signature in particular. We're not there yet, but the pillars of the analysis are there, and we're about to share them with the community to get their feedback. Okay, with regards to these advances as far as research with the implementation of new technology, you're creating a tool for a matrix construction. Do you have any implementation of that technology for this artificial origin analysis? Well, the reality is that we don't yet have the tools that we need. These tools needs to mature a little bit more. This is a project that we have just started with Axel and a very close partner in the Mathematics Institute in Michoacan, and we're barely starting to create the first concepts. To create functional analysis, we require genomes that are, we need 5 times of what we have right now to generate tools of neuronal algorithms. We require of more samples. And the thing is, we don't want to feed these algorithms with genomes from databases because we know that they do not necessarily represent the region. So we can, but we can start drawing the concepts, and that's what we're starting with. And we have started working with those algorithms so that all of the genomes that are generated from now on would fit that model that would be not exclusive, but that would cover the particularities of the lineages in the region. Thank you, Doctor. I see that Dr. Roberto Zenteno has his hand raised. Go ahead, Doctor. Yes, I'm sorry to interrupt, but there was a question about autochthonous lineages, and I wanted to mention something about this. Unfortunately, in Mexico, even though we have a great tuberculosis burden, the genomes that have been sequenced are very few. And that allows for us to do certain analysis, but we cannot do it at a more detailed level. So with the analysis, the genomic analysis that we have from Veracruz, we were able to come up with an X3 lineage, which is exclusive for Mexico and that has not been reported in any other parts of the world. And this on top of this lineage, this is a lineage that has been circulating for 10 or 15 years in Mexico, and it has a big tendency to create drug resistance. And we have been able to correlate these lineages with resistances, and we were able to track them in the past. Now, when we start getting more information, we're going to be able to start analyzing more lineages that are typical for Mexico, including some that are specifically for certain geographical regions, because these differ from the north part to the south part of our country. And once we have this, we will have a better overview of the different lineages in our country, especially as far as the drug resistance that each one of them generate. And this will be information that would be very valuable and we hope to have in the near future. Okay, our last question due to the time, those molecular research, what did they give as far of creating an effective vaccine? Well, as far as creating an effective vaccine, well, this type of analysis, this type of functional analysis are not, well, the impact is not direct on a vaccine. Perhaps what comes up to mind, and somehow it is the type of results that those analyses give us is they give us a target. They give us well, not finding it, but what this analysis do is they propose targets under this cellular program that has been affected on these type of lineages or on these types of samples, and then we can try to generalize it or we can focus it on a particular target. A vaccine has nothing to do with the phenotypes and with those kinds of issues. That's why there's not a direct relation. And even though it gives important comparative information. Perhaps some of these metabolic models where you can prove viability, which are models that come from the genomic sequencing and can create emergent mapping, then you can say, well, this strain with these mutations, is it possible or not possible? That could allow for us to find a target to then develop a vaccine. Well, thank you so much, Doctor. With this, we finish the Q&A session. We thank everyone who participated on this session and to make it possible. And we also thank our interpreters today. Please register your QR code so that you can get your certificate for participating on this 4th session. And we invite you to be a part of the 5th session of the Tuberculosis Forum next Thursday, November 11. Dr. Orango from the OBS and is going to talk about childhood tuberculosis, as well as Dr. Lucia Alvarez Hernandez, who also works for a clinic for specialized on children and teenagers at a clinic in Mexico. Thank you so much for being here today. Thank you, everyone.